Hello everyone, I am Payal Priyadarshini, PGT Biotechnology from Kendriya Vidyalaya number 3 Delhi Cant. In my last session, we learned about protein synthesis, that is translation, means how messenger RNA codes for amino acids and how those amino acids combines together to form a polypeptide chain, or we can say protein. So, in that session we learned about the different labels of a structure of proteins, the different bonds which involve in the protein and the different steps involved in the synthesis of protein. There are 4 steps. The first one is the charging of uncharged transfer RNA, that is binding of amino acid with the uncharged transfer RNA. Second step is initiation of protein synthesis. Third step is elongation of protein chain. And the fourth step is termination of protein synthesis. In the last session, we learned about charging of uncharged transfer RNA, in which amino acid transfer RNA synthetase helps in the binding of amino acid with the uncharged transfer RNA to form charged transfer RNA to initiate the translation. Before going more in detail in this topic, first let us get the answer of the questions which we asked in my last session. The first question was, what do you mean by protein synthesis? Protein synthesis means production of protein. So, formation of polypeptide chain or protein from messenger RNA inside the cell is called protein synthesis or translation. Next question was, which enzyme helps in charging of transfer RNA? Charging of transfer RNA means binding of amino acid with the uncharged transfer RNA, which will be bring up to the messenger RNA during protein synthesis. So, the enzyme is amino acid transfer RNA synthetase. This enzyme help in the charging of uncharged transfer RNA, that is binding of amino acid with uncharged transfer RNA. The next question was, which codon is called initiation codon? Yes, the answer is right, AUG. AUG is considered as initiation codon which always code for methionine. The next question was, what do you mean by stop codon? Stop codon, as we know, there are 3 stop codons, UAG, UGA and UGA. So, stop codon gives signal to terminate the protein synthesis. When the protein synthesis is being started, how it is being terminated? So, in that termination the stop codon helps. The next question was, what are the role of ribosomes? Very important question. So, ribosomes is one of the cell organelle which help in protein synthesis. During protein synthesis, ribosomes recognizes the initiation site for protein synthesis. It also reads the codon of messenger RNA and it also helps in termination of protein synthesis. So, these are the different function of ribosomes inside the cell during protein synthesis. Now, let us start today's session, that is initiation of protein synthesis, how the protein is being initiated in the cell during the process of protein synthesis or translation. The initiation of protein synthesis requires ribosomes, a specific initiated charge transfer RNA, messenger RNA, GTP as energy source and some initiation factors in case of prokaryotes. If we talk about ribosomes in case of bacteria or prokaryotes, the ribosomes have 2 subunits. One is larger subunit, that is 50S, and second one is a smaller subunit, that is 30S. The ribosomes contain 23S, 5S and 16S ribosomal RNA, and these ribosomes also reads the codon of messenger RNA. By reading the codon of messenger RNA, it gives the signal to the transfer RNA to bring the amino acid during protein synthesis. So, this is about bacterial ribosome. if we talk about eukaryotic ribosomes, in case of eukaryotes, they have also 2 subunits like prokaryotes, but in case of eukaryotes, the largest subunit is of 60S and a smaller subunit is of 40S, and the ribosomes contain 80S, 5.8S, 28S and 5S ribosomal RNA. And like that of prokaryotic ribosome, it also reads the codon of messenger RNA for the protein synthesis. So, this is a typical structure of ribosome you can see in the slide. This one is a prokaryotic ribosome, smaller subunit, larger subunit. When it will combine, it will give the complete ribosome. A smaller subunit of prokaryote is of 30S and larger subunit is of 50S. After combining, they will form 70S ribosome. In case of eukaryotes, a smaller subunit is of 40S, larger subunit is of 60S. After combination of these subunits, they will form 80S ribosomes in case of eukaryotes. So, if we talk about ribosome, the ribosome have 3 specific sites for protein synthesis, and these sites are A site, that is amino acid site. At A site, the charge transfer RNA always combines and bring up the amino acid. P site, that is peptidyl site, this site brings or contains the polypeptide chain, and the third site, that is E site, is the exit site from where the uncharged transfer RNA leaves the complex. So, these are the 3 main important sites of ribosomes which helps in protein synthesis. The first one is A site, that is amino acid site, second one is P site, that is peptidyl site, third one is E site, that is exit site. Now, let us talk about initiation factors in case of prokaryotes. There are 3 initiated factors. The first one is IF1, second one is IF2 and third one is IF3. So, these are the 3 initiation factors which involve in the initiation of protein synthesis in prokaryotes. Initiation factor 1 blocks A site for ribosome. As we learned कि ribosome is having 3 sites, A site, P site and E site. A site is being blocked by IF1 during the initiation of protein. Second one is IF2, that is initiation factor 2, which brings GTP molecule as an energy source. And the third one is initiation factor 3, which prevent association of larger subunit of ribosomes with a smaller subunit. During initiation, first of all, the smaller subunits will combine with the different components to form the 30S initiation complex. During this activity, the larger subunits cannot bind, and it cannot bind because of the IF3, that is initiation factor 3, which prevents the association of larger subunits with the smaller subunit. Now, steps of initiation in prokaryotes. There are 3 basic steps. The first one is messenger RNA binds to a small subunit of the ribosome. So, first of all, the messenger RNA will bind with smaller subunit of ribosome. Secondly, the initiated transfer RNA binds to messenger RNA through base pairing between the codon and anticodon. And third step is the large subribosomes joins the initiation complex. So, these are the basic 3 steps of the protein initiation, and these steps are also subdivided in different substages. So, these steps are messenger RNA binds to the smaller subunit of the ribosome, initiated transfer RNA binds to the messenger RNA through base pairing between the codon and anticodon. The larger subunit joins the initiation complex to form the 70S initiation complex to initiate the protein synthesis. Now, let us talk about how messenger RNA will binds with smaller subunit of the ribosome. Now, binding of messenger RNA to the small subunit of the ribosome involves different substages. The first one, the binding of IF1 and IF3 with a smaller subunit of ribosome. That means, first of all, as we talk about initiation factors, there are 3 initiation factors. Out of these 3, only 2, that is IF1 and IF3 will bind with the smaller subunit of ribosome. After the binding of IF1 and IF3 with the smaller subunit, the ribosome binds at AUG codon. AUG codon is initiator codon which initiate the protein synthesis of messenger RNA. And after that, Shine-Dalgarno consensus sequence which is there in the messenger RNA, and this sequence is complementary to a sequence of nucleotide at the 3 end of 16S ribosomal RNA, which also helps in the binding of ribosome with the messenger RNA. So, these are the different steps of how messenger RNA binds to a small subunit of ribosome. Next step was initiated transfer RNA binds to the messenger RNA through base pairing between the codon and anticodon. This step requires IF2, that is initiation factor 2. This initiation factor 2 forms complex with GTP molecule as energy source. IF1, that is initiation factor 1, enhance dissociation of larger and a smaller subunit. Initiation factor 2, when combined with GTP, it will form IF2 GTP complex along with formyl methionine transfer RNA binds to form 30S initiation complex. So, IF1, IF2, IF3 along with GTP molecule, messenger RNA, formyl methionine transfer RNA, that is initiated transfer RNA and messenger RNA with 30S smaller subunit of ribosome, they all will combine to form 30S initiation complex. This we can learn in this diagram also. Here you can see, first of all, with this 30S ribosomal subunit, that is a smaller subunit of ribosome, IF1 and IF3, the two initiation factor which helps in protein synthesis, will binds with 30S ribosomal subunits. After the binding of initiation factor 1 and initiation factor 3, this messenger RNA which have the Shine-Dalgarno sequence, this Shine-Dalgarno sequence is the complementary of the sequences of the ribosomal RNA which is there in the ribosome. With the help of this complementary nature, this 30S ribosomes will bind with the messenger RNA. And after the binding of messenger RNA with the smaller subunit and initiation factor 1 and 3, the charge transfer RNA, the first charge transfer RNA which is going to bind is formyl methionine amino acid transfer RNA. This charge transfer RNA with the help of GT molecule will combine with this complex to form 30S initiation complex. Here you can see the messenger RNA having AUG codon as the initiation codon, this AUG having the anticodon UAG in the charge transfer RNA, and as AUG code for methionine, this charge transfer RNA will bring formyl methionine amino acid in case of prokaryotes, and this whole complex is called 30S initiation complex. After the formation of 30S initiation complex, the larger ribosome, the larger subunit of ribosome joins to the 30S initiation complex, and this involves IF3 dissociates from complex, the initiation factor 3, because initiation factor 3 prevent the association of 30S and 50S subunits of ribosome. So, if IF3 will remain there in the complex, there will not be association of a smaller subunit with the larger subunits. So, for the association of larger subunit with smaller subunits, the IF3 factor has to be released. So, this IF3, that is initiation factor 3, will dissociate from the complex. Larger subunit, that is 50S, binds with smaller subunit. This requires energy, and energy is being given by the GTP molecule. This GTP molecule hydrolyzed to form GDP and gives the energy. After this, the initiation factor 1 and initiation factor 2 depart from the complex. And now after the departure of IF1 and IF2, the formation of 30S initiation complex is there, which will help in the initiation of protein synthesis. You can understand much more in detail with the help of this diagram. Here you can see this one is 30S initiation complex. This one is 30S initiation complex. This 30S initiation complex have a smaller subunit, that is 30S with initiation factor 1 and 2 with charge transfer RNA having formyl methionine as amino acid at the 3 end along with the GTP molecule. Now with this 30S initiation complex, this larger subunit, that is 50S ribosomal subunits, will combine, but it will combine only when the initiation factor 3 will dissociate from this complex. So while combination of this 50S ribosomal subunits with this 30S initiation complex, all the initiation factors will depart along with the hydrolysis of GTP in GDP with the release of energy, and that energy is utilized in the binding of 50S with the 30S subunit of ribosomes. When 50S ribosomal subunits combine with 30S initiation complex, they will form 70S initiation complex. This 70S initiation complex contains a smaller subunit of ribosome, larger subunit of ribosome, initiator transfer RNA and messenger RNA. Now this 70S initiation complex will start the protein synthesis. From here the protein synthesis will be initiated. If we talk about the steps of initiation in eukaryotes, it is somehow similar to the prokaryotic initiation, but with some differences, and these differences are eukaryotes requires eukaryotic initiation factors. In case of prokaryotes, the prokaryotes needs initiation factors 1, initiation factor 2 and initiation factor 3. But in case of eukaryotes, the eukaryotes requires eukaryotic initiation factors, that is EIF. Initiator methionine is unmodified. In case of prokaryotes, the first amino acid which is going to bind with the polypeptide chain is formyl methionine, which is modified amino acid. This type of modification is not there in case of eukaryotes. Shine-Dalgarno sequence not found in the messenger RNA. As we know, messenger RNA of prokaryotes have Shine-Dalgarno sequence which helps the ribosome to recognize the initiation codon. This type of Shine-Dalgarno sequence is not there in eukaryotic messenger RNA. Eukaryotic initiation factor 4E cap binding protein, that is CBP, binds to cap at 5 end. As we know, in case of eukaryotes, at the 5 prime end there is cap. Cap means there is unusual 5 prime binding of 7-methyl guanosine triphosphate with the 5 prime end of the messenger RNA. So to initiate the protein synthesis in case of eukaryote, eukaryotic initiation factor 4E cap binding protein binds to cap at 5 prime end. Complex of 40S ribosomal subunit with initiator methionine transfer RNA and several eukaryotic initiation factor and GTP binds together and moves along the messenger RNA to find the AUG start codon. So it is somehow different from the prokaryotic. In case of prokaryotic ribosomes, with the help of Shine-Dalgarno sequence, they can recognize the initiation codon, but in case of eukaryotes, the 40S ribosomal subunit with initiator methionine transfer RNA with several several eukaryotic initiation factor and GTP binds together and moves along the messenger RNA to bind AUG start codon. Now, let us talk about elongation of protein chain. We learned in earlier slide how 70S initiation complex is being synthesized to initiate the protein. After the binding of first amino acid at the P site of ribosome, how the further amino acid will combine, that is elongation of protein chain. The elongation of protein chain requires 70S initiation complex, which already have formed in initiation of protein synthesis, charge transfer RNA with their amino acid, elongation factor and GTP molecule as energy source. So, these are the requirements for the elongation of protein chain. Elongation occurs in 3 different phases. The first is delivery of charge transfer RNA to the A site. As we know, A site is for the binding of charge transfer RNA during protein synthesis. So, A site is always free for the binding of charge transfer RNA during protein synthesis. The first step of elongation is delivery of charge transfer RNA to the A site. Second step is creation of peptide bond between amino acid. As we know, for the formation of polypeptide chain or protein, the amino acids have to combine together, and they combine with the help of peptide bond. So, in the second step of elongation, the amino acid will combine with the help of peptide bond. And the third step is translocation. Translocation means the ribosome will move one triplet codon towards the 3' end of messenger RNA. So, these are the 3 different phases of elongation of protein chain. Now, these different phases are also have different substages, which we discuss in coming slide. So, the first step is delivery of charge transfer RNA to the A site. That means how the incoming charge transfer RNA will combine to the A site, which is free for the charge transfer RNA. It requires elongation factor EF-TU. TU stands for thermo unstable. Elongation factor TS. TS stands for thermostable, and GTP molecule as energy source. Elongation factor TU joins with GTP and then binds to a charge transfer RNA. Next to this, when elongation factor TU joins with GTP binds to the charge transfer RNA, they will form 3-letter complex, and this 3-letter complex enter the A site of ribosome. Anticodon of transfer RNA pairs with messenger RNA. Whatever codon is there in the messenger RNA, accordingly the anticodon transfer RNA will come and bind complementarily with the codon of the messenger RNA. During this process, GTP cleaved to GDP to release the energy for this process. Elongation factor TU GDP complex is being released after the combination of charge transfer RNA with the messenger RNA, and then again this elongation factor TU GDP regenerates into elongation factor TU GTP, so that the process can be cycled. So, this we can understand with the help of this diagram. So, you can see here in this diagram, this one is 70S initiation complex, 70S initiation complex. With this 70S initiation complex, you can see in the diagram, at P site always the first charged transfer RNA will combine, and A site is always free for incoming charged transfer RNA. So, the incoming charged transfer RNA will bind at A site with the help of this cycle. You can see in the cycle, as you can see in the diagram, UCC is the next codon which will code for the serine. So, the anticodon for UCC is AGG. With this transfer RNA having anticodon AGG, serine will bind in the same process as we learned in the charging of transfer RNA. Now, this charged transfer RNA will combine with elongation factor TU and GTP complex. You can see in the diagram. This GTP elongation factor TU will combine with charged transfer RNA to form charged transfer RNA elongation factor TU GTP complex. Now, this charged transfer RNA which is combined with elongation factor TU and GTP, from here the elongation factor TU and GTP molecule will be released, and this charged transfer RNA will combine at the A site of the 70S initiation complex. You can see in the diagram, at A site there is next charged transfer RNA with its amino acid. We talk about this cycle is called elongation factor TU-TS exchange cycle. You can see over here, after the release of elongation factor TU and GTP, this GTP will be hydrolyzed to form GDP and one phosphate will be removed. Now, this complex elongation factor TU and GDP molecule, with this complex TS factor will be added up, and after the addition of this TS factor, this GDP will be released, and now the combination of elongation factor TU-TS complex will be synthesized. With this elongation factor TU-TS complex, the GTP molecule will attached. After the binding of this GTP molecule with this complex, the TS molecule will be removed, and when this elongation factor TU GTP molecule is being synthesized, with this complex the charge transfer RNA will be added up, and then it will help in the binding of this charge transfer RNA at the A site of the ribosome. Now, you can see in this particular diagram there are two amino acid, one is formylmethionine and second one is serine. So, this is how the charge transfer RNA will combine with the A site. Now, let us see how these two amino acid will form the peptide bond to elongate the protein chain. The second step of elongation, that is creation of peptide bond between amino acid. Peptide bond forms in between amino acid attached to P and A site. You can see in the diagram, we have two amino acid attached with charge transfer RNA at P site and A site. Next step of this peptide bond formation, amino acid released from P site from its transfer RNA. This P site charge transfer RNA from this site, this amino acid, methionine amino acid will be released. When it will release, it will form the peptide bond with the adjacent amino acid. Activity is catalyzed by peptidyl transferase, that is the transfer of this formylmethionine from P site to A site for the formation of peptide bond is catalyzed by an enzyme which is called peptidyl transferase enzyme. You can see in the diagram, this amino acid which is there in the P site will be released by the enzyme peptidyl transferase, and it will combine with the next amino acid to form peptide bond to form polypeptide chain. You can see in the diagram. From this amino acid, this amino acid will form a peptide bond, and like that the chain will be elongated. Now, the third step of elongation is translocation. Translocation means movement of ribosome down the messenger RNA in the 5 prime to 3 prime direction. Translocation means this whole ribosome will move one triplet codon towards the 3 prime of the messenger RNA. You can see in the diagram, in this particular diagram, this ribosome having A site with UCC codon of messenger RNA and P site with AUG codon of messenger RNA. When this whole ribosome will move one triplet codon ahead towards the 3 prime end of messenger RNA, you can see after the movement of the ribosome, which is called translocation, the P site will come at E site, the A site will come at P site, and the A site will be empty for the incoming charge transfer RNA. And at the P site, this 2 amino acid will be there with the peptide bond. You can see in the diagram, after the translocation of this particular ribosome, you can see here AAG codon is there in the messenger RNA, which is not there inside the ribosome. When this ribosome will translocate in one triplet codon towards the 3 prime end, this AAG will come in the A site. Here you can see this AAG is there in the A site of the ribosome. At the P site, there is 2 amino acid combined with the peptide bond, and this particular transfer RNA, when it will release its amino acid, it become uncharged. You can see in the diagram, this one is uncharged transfer RNA because it has given its amino acid to the adjacent amino acid, and this uncharged transfer RNA will release from the E site. So, this is how the translocation occurs inside the cell. Translocation requires elongation factor EFG. You can see in the diagram here the movement of this ribosome one triplet codon ahead requires elongation factor G. Elongation factor G with the help of GTP molecule will form elongation factor G GTP complex, and this elongation factor G with GTP complex will help in the translocation of ribosome one triplet codon ahead towards the 3 prime end, and after the translocation, this elongation factor G will be released along with the release of GTP molecule. GTP molecule is used as energy resource. So, this GTP molecule will give the energy and it will hydrolyze in GTP. Now, this GTP molecule will combine with the phosphate to form GTP molecule, and the cycle will goes on like this. So, in the translocation of ribosome, elongation factor G helps. Hydrolysis of GTP to GTP, this we learned in the slide. This causes A site free for receiving the charge transfer RNA. You can see due to translocation of this ribosome will give free A site, so that the incoming charge transfer RNA will combine over here to elongate the protein chain. Now, the last step of protein synthesis, that is termination of protein chain or protein synthesis. How the protein chain will be terminated once it has been started, once it has been initiated and elongated, how it will be terminated. This termination requires stop codon. As we learned in the genetic code, there are 3 stop codons which do not code for any amino acid, and these are UAG, UGA and UAA. These are 3 stop codon: UAG, UGA and UAA. stop codon do not code for any amino acid. These stop codon UAG, UGA and UAA do not code for any amino acid, that is why they are also known as nonsense codon. Ribosome recognize stop codon with the help of protein called release factor. When this ribosome will move ahead and ahead towards the 3' end, the chain of this amino acid will be elongated. When this ribosome will reaches to any of these stop codon, this stop codon will help in the binding of release factor to the A site. There are 3 release factor in case of prokaryotes: release factor 1, release factor 2 and release factor 3. These 3 release factors helps in the termination of protein synthesis in case of prokaryotes. Release factor 1 recognizes UAA and UAG. This release factor 1 will help in the recognition of UAA as well as UAG stop codon, and release factor 2 recognizes UAA and UGA stop codon. So, recognition of a stop codon is initiated by release factor 1 and release factor 2. Binding of release factor with the stop codon triggers peptidyl transferase to cleave the polypeptide from transfer RNA in the P site. Let us have a look on the diagram for the termination of protein synthesis. This is the whole diagram for the termination of protein synthesis. You can see at the P site, this is a polypeptide chain which have already been synthesized by the elongation of protein chain. Now, this ribosomes has reached to stop codon. Here we have UAG stop codon. Now, when the ribosome will reach to the any of the stop codon, the release factor will be added up, and this release factor will trigger peptidyl transferase enzyme. This is very much important, peptidyl transferase enzyme, to cleave the polypeptide from transfer RNA in the P site. You can see the polypeptide chain is there in the P site. When the ribosome will reach to the stop codon, the release factor will be bind at this A site, and after the binding of release factor, this release factor will give trigger to the peptidyl transferase to release this polypeptide chain from P site. Next to that, polypeptide chain leaves the ribosome. When peptidyl transferase will remove this polypeptide chain from P site, this polypeptide chain will leave the whole this ribosome and will reach to the cytoplasm. Release factor 3 along with GDP binds to the ribosome. This stimulates the releasing of release factor from stop codon and ribosome. When release factor 3 with GTP molecule binds with the ribosome, the combination of the release factor 3 with GDP, it stimulates the releasing of release factor from stop codon and ribosome. Now, GTP replaces the GTP. This GTP which is attached with the release factor 3 will be replaced by GTP molecule. Release factor 3 hydrolyzes GTP, which allows release factor 3 to be released from the ribosome. Release factor 3 hydrolyzes GTP molecule, and hydrolysis of GTP molecule will help the release factor 3 to get released from the ribosome. Now, after the release of release factor 3 from the complex, how the whole ribosome complex will be dissociated, or we can say deconstructed, that is deconstruction of ribosomal complex. So, before that, let us understand this diagram. You can see at the P site here we have polypeptide chain. A site is having UAG as a stop codon. The ribosome will trigger release factor 1 to combine at A site. After the combination of release factor at the A site, it will trigger the peptidyl transferase enzyme, and the peptidyl transfer enzyme will break the bond in between transfer RNA and polypeptide chain, and because of the breaking of the bond, this polypeptide chain will be released from the ribosome and it will reach to the cytoplasm. Now, with the whole this complex, the release factor 3 and GDP, guanosine diphosphate, you can see here release factor 3 and GTP will combine. The combination of this release factor 3 GDP complex with this ribosome will help in the release of this release factor 1. When the release factor 3 GDP molecule will combine with the ribosome, the release factor 1 will be released from ribosome. With the release of this release factor 1 from the ribosome, this release factor 3 will also get released from here, and this GDP will help the binding of ribosomal recycling factor at A site. From here the ribosomal recycling factors will be transferred to the P site, and from P site the elongation factor G with GTP molecule will combine, and the whole this factor will be dissociated separately from each other. The messenger RNA, 30S complex, 50S ribosome, uncharged transfer RNA, elongation factor G, GTP molecule and ribosomal recycling factor. All these factors will be dissociate from each other, and then the protein synthesis will be stopped, or we can say terminated. So, deconstruction of ribosomes require ribosomal recycling factor. Here you can see it is ribosomal recycling factor. This ribosomal recycling factors binds to A site. As we know, A site is free. At this A site, ribosomal release factor will combine. Now, elongation factor G binds, which causing translocation of ribosome. With this complex, elongation factor G will be attached, and the binding of elongation factor G, this whole ribosome will move one triplet codon ahead, that is translocation of ribosome will be there. Ribosomal recycling factor moves to the P site and uncharged ribosomal RNA to E site. You can see when this whole particular ribosome will move one triplet codon ahead, this uncharged transfer RNA will reach to the E site. This ribosomal recycling factor will reach to the P site, and A site, at the A site elongation factor G will be there. Ribosomal release factor releases uncharged transfer RNA. This ribosomal release factor will help in the release of this uncharged transfer RNA from the ribosomes. Elongation factor G releases ribosomal recycling factor. This elongation factor G will help in the release of this ribosomal recycling factor from the ribosome. You can see this uncharged transfer RNA has been released, ribosomal recycling factor has been released, and then both subunit of ribosome dissociates from each other. Both the subunit 30S and 50S, both will be released separated from each other. So, we can see how the protein is being initiated with the help of initiation factors, how it is being elongated with the help of elongation factors, and how it is being terminated with the help of termination factor, or we can say release factors. So, it is a very important to note that there are 3 basic steps for protein synthesis. One is charging of transfer RNA. Charging of transfer RNA means the amino acid will combines to the uncharged transfer RNA to form charged transfer RNA, and this will be catalyzed by the enzyme amino acid transfer RNA synthetase. This catalysis is always there inside the cell for every transfer RNA depending on its anticodon. Next to this, the protein synthesis is being initiated with the help of initiation factors. In case of prokaryotes, there are 3 initiation factors: IF1, IF2 and IF3. In case of eukaryotes, there are so many initiation factors are there, which are known as eukaryotic initiation factors. These initiation factors helps for the formation of 70S initiation complex, so that the protein synthesis can be initiated. After the formation of 70S initiation complex, the elongation factors will involve for the elongation of protein chain. Here we need to learn how the amino acid will added up with the uncharged transfer RNA. This we have already learned in the charging of transfer RNA, but with the help of which bond the amino acid will combine with the charged transfer RNA. This is a very important concept which we should know. So, you can see in this diagram, this one is an uncharged transfer RNA, this one is uncharged transfer RNA. Uncharged transfer RNA means this transfer RNA is not having any amino acid with it. With this uncharged transfer RNA, say suppose this amino acid going to bind. My question is, how this amino acid will combine with this uncharged transfer RNA? As we know, the binding of amino acid with the uncharged transfer RNA is catalyzed by an enzyme which is known as amino acid transfer RNA synthetase. This amino acid helps in the binding of this amino acid with the uncharged transfer RNA, but here you need to learn how the bond is being formed. You always remember that at the 3' end in the RNA there is OH group, hydroxyl group, and in case of amino acid, this is an amino acid, here it is an R group, this is amino group, and this one is carboxylic group. You always remember that this carboxylic acid, the C end of the amino acid, will always interact with the 3' end of the RNA to form the bond, so that it can combine at the 3' end. How the bond is being synthesized? This OH of the acid and this hydrogen of the RNA will interact together to form the water molecule H2O. Now, this H2O will be released from here, and this amino acid will combine with the 3' end with the covalent linkage. So, you always remember that the amino acid will always combines at 3' end of the charged transfer RNA, and at the 3' end always the C part, the C end, the C terminal of amino acid will combines. So, the protein synthesis, if we talk about the direction of protein synthesis, as we know in a particular amino acid, so suppose it is an amino acid is having C terminal, N terminal, R group, R may be any group, and hydrogen. So, the direction, the important question, direction of protein synthesis. What will be the direction of protein synthesis? Whether it will be N to C or C to N. N means N terminal, means the protein having the amino group, and C terminal means the protein having the carboxylic end. So, what will be the direction of protein synthesis? Whether it will be from N to C or C to N. So, you can see the C, the carboxylic part of the amino acid is combining with the transfer RNA. So, the nitrogen part, that is N terminal, this N terminal is always free, and C part will always combine with the transfer RNA. So, the direction of protein synthesis is always and always N terminal to C terminal. That means, if there is a chain of amino acids, say suppose this amino acid has been combined with another amino acid, say A2, A3, like this. So, this side there is always C terminal, and in the here we have always N terminal. So, direction of protein synthesis is always N to C, amino group to carboxylic group. So, this is how the protein synthesis is being synthesized, protein is being synthesized inside the cell with the help of different proteins, different elongation factors, initiation factor and release factor. Here I am going to stop my session. In my coming session, we will discuss some of the questions related to today's session, and then again we will learn for the regulation of gene expression. Till then, thank you, have a nice day.